Good afternoon to everybody, and a big thank you to TBS BioCon for giving me this opportunity to talk. Um, what I want to start off with is I'm right now affiliated with SUNY Downstate Medical Center, but this talk will basically be around the topics that I've worked on during my PhD in IIT Bombay and cellular communication, as I hope the title is familiar, um, rings a bell, is basically how cells communicate or what kind of electrical signals they use to communicate to one another or across multiple levels. So, uh, oops, yeah. Um, so my profession is I'm researching in the field of computational neuroscience. So neuroscience by definition is basically the study of neurons, but right now the scope has really increased. That it's more broad than just it's broader than just looking at brains and neuronal networks, because initially that is how it started, but now it looks at brain neuronal networks, how neurons affect other organs, then other organs by themselves, for instance the heart, the bladder, which was my PhD topic. Actually, I worked on the bladder, then the pancreas, the gut, for instance, a lot of work going on even on the gut. So now neuroscience has become, on a more broad perspective, it is basically anything that will help you understand your body better, or anything that will help you diagnose or help with prognosis, treatment, identification of pathologies, pathophysiologies. And so as the name or as the description is, you can't look at these things at only one level. So neuroscience actually spans multiple levels. It maps onto, for instance, the whole organ. For instance, if you're looking at the image at the side, so you have something like the whole central nervous system that the brain controls, or you look at systems at a system level, or you look at maps, or networks, network of pyramidal cells or network of some other cells, and how they interact with each other, just localized bits. Then you go much deeper and you look at how each individual cell will probably behave, or how neuron, one axon to another axon, how do they connect, synapses. And if you go further down, you're actually looking at signaling molecules. So all of these now comprise of research in any of these fields come under neuroscience, with the perspective, of course, of understanding how the body is. And because you're spanning such a wide range, if you look at this is like angstrom level to about meter level. If you're spanning such a wide range, you also need different techniques, experimental techniques, computational techniques, computational power. So all of these now, is something, and it all depends on the question you're answering. If you're interested to know how a molecule is being is moving from, how it is used as a signaling, in a signaling cascade, then you need a different set of questions, you need a different set of experimental techniques, computational resources. All of those change. So neuroscience was actually around from 1900s. So I don't know how many of you know this, but Jagdish Chandra Bose from India, he actually published quite a few papers in the early 1900s, which in which he studied about plant cell excitability, as in how plant cells communicate their action potentials, plant action potentials, which is actually very interesting. These papers are online if any of you would like to take a look. um, okay. So surface recording of signals, these bioelectric signals that we are most comfortable with, any layman, anyone with even the slight one visit to the hospital will probably have an idea about what ECG is, EEG is, EMG is, EOG, ERG, oculogram, retinogram, gastrogram. Because if you have a problem, suppose you go in with some sort of a heart ailment or something, the first thing the doctor will do is probably check your ECG and see that the basic trace is normal. But the thing is that our body, how does it generate these electrical signals all across? So in the brain, in muscle, in the heart. So how does it generate in the eye, in the gut? How is it generating? Is there are there signals that are more fundamental that we are not recording right now, or in the hospitals, but they are present, and a spatio-temporal integration of those signals is what gives rise to these macro signals. So the answer is yes. So there are cellular signals. Each cell in our body, they have something known as a resting membrane potential. They are already at some sort of a negative potential by default. Then you have presynaptic, postsynaptic. Here, synapse, I'm using it in a loose way. It can also mean neuromuscular junction. So, across between two nerves, between a nerve and a muscle, when they send signals out, you have an electrical signal that's produced. And the one that will be the highlight of this talk will be action potentials. Action potentials are actually a proper electrical signal, a millivolt electrical signal. I'll get into the details. And it can travel from the brain all the way to the tip of the toe at constant amplitude and without any attenuation. So it's a non-lossy signal, and it's most optimum for uh, uh, propagation. And that is how most of our signals, so for instance, right now, if you're reaching out to grab a glass of water, or you're reaching out to mute something or increase the volume, all of these things are being governed at a cellular level by your action potentials. So even if you don't know there are action potentials, while you're listening about action potentials, there are like a million action potentials going on in your brain, firing in your brain. So this is a very rough cartoon of how these signals, the underlying machinery of these signals, because after this, these things will have some importance, and that's why I thought it's better to kind of set the stage. So when the cell is unperturbed, no stimulus, nothing, then the cell is at its RMP, which is the resting membrane potential, which is shown here. Different cells have different resting membrane potential. Your smooth muscle of the bladder versus skeletal muscle versus cardiac versus neuron, all of them will have different RMPs. And these RMPs are basically controlled by your leak channels, by your pumps, and by your exchangers in the membrane. Now, suppose there is a perturbation, some sort of a stimulus. I'm assuming here from a neuron. Then if it the neuronal receptors, I mean the neurotransmitter receptors, they could be inotropic or they could be metabotropic. So if you have inotropic receptors, then there's an inflow of ions, and there is a slight second yeah and there is a slight depolarization. Now, these depolarizations are actually between nerve to nerve or nerve to muscle. These are known as presynaptic, postsynaptic potentials. These are subthreshold signals. That means these do not cross a particular threshold that's very unique to the cell. And why is the threshold important? I'll come to that again. Metabotropic receptors are another class of receptors, not getting into this, but these receptors don't have anything to do with an ion influx across the cell, and therefore they just trigger off intracellular signaling cascades. So once now, if there is an influx of ions, an efflux of ions, both influx and efflux of ions, such that, and the influx is higher than the efflux, so an influx of positive ions will raise the membrane potential progressively, and if it can cross the threshold, then an action potential is fired. Now, what happens during this action potential? Basically, when the membrane potential crosses a threshold, calcium channels and sodium channels, voltage gated calcium and sodium channels, these channels are protein structures that span across the transverse of the membrane. So they are at one end, they are connected to the extracellular fluid, and the other end they are in contact with the intracellular fluid. So once these two activate, they actually result, because they are allowing an influx of calcium ions, which is 2 plus, an influx of sodium ions, which is 1 plus. It actually allows the potential to increase. Or the same thing, this is a cardiac action potential, and this one also allows the potential to increase. So on two sides, I have two prototypical action potential, one for the nerve and one for the cardiac. In due course of time, for the cardiac, the calcium active, the voltage activated potassium channel, the K channel, also activates, and these channels actually allow the potassium ions to move out of the cell. And when that happens, then suddenly during in the cardiac action potential, there is some sort of a balance achieved between the calcium channels and the potassium channels. So there's some sort of a plateau phase here. But in general, After that, the calcium channels switch off. Sodium channels switch off much earlier, but channels switch off soon after that. And when potassium channels take over, they actually bring the potential back to the resting state. In fact, sometimes even lower than the resting state, because they are allowing an efflux of potassium ions. So now the positive ions are moving out of the cell. So each of these potentials will come out, and then in due course of time, once the voltage membrane potential falls down, decreases, repolarizes, the potassium channels switch off, and again the pumps and exchanges kind of take over, and RMP is restored again. So now this is how an action potential is elicited in a single cell. But how is it propagating? So in terms of a nerve propagation, so if you have an action potential that's elicited at the soma or at one of these axon hills, what this is image is trying to show is that in an unmyelinated axon, so action potential has, so in our body we have myelinated axons and unmyelinated axons. So in unmyelinated axons, which is just the native axon, just the raw axon, it has a dense concentration of sodium and potassium and whatever calcium ion channels, and one perturbation or one stimulus here, and the action potential will just flow through. This will be activated, then the next set of ion channels will be activated, then the next set of ion channels will be activated, and so on and so forth. But these unmyelinated axons are actually slower in terms of action potential propagation. And because at each point action potential is being separately triggered, each of these amplitudes stay the same. And till the end of the axon, the action potential has propagated right from this soma all the way till the end. But these axons, because it's stimulating an action potential every minute, at every unit space, unit area, these things are actually much slower. In myelinated axons, these axons conduct action potentials much faster. And how do they do that? Myelinated axons basically have small, sorry, not small, but largish gaps in the largest portions of the axon are wound by myelin sheath. Now, these myelin sheath do not have ion channels of their own, and they do not, and because they are covering the axon, there are no ion channels present underneath the myelin sheath. So here what happens is that at each gap, which is known as the node of Ranvier, at each gap, there is a dense concentration of ion channels. So once there's an action potential fired at one of the nodes of Ranvier, there is a passive propagation across the myelin sheath, across the region that is encapsulated by the myelin sheath, till the point it reaches the next node of Ranvier. Now here one needs to remember that, again, which is why I use the word cellular communication, that it's very similar to our transmission lines. So at every point in our transmission lines, you need a repeater, you need a booster. So think of it like this, that these nodes of Ranviers are basically boosters to the electrical signal. And this is a passive thing, which it does not have ion channels, and therefore there's a passive conductance, okay, passive flow of ions here, which makes action potential propagation much faster, because it's only getting triggered at these gaps. But now the thing is that even in electrical transmission, if you have a big enough gap and you don't have repeaters or boosters at every interval, and there's a big gap, suddenly a booster is not working, the potential just, it falls, it decreases. And that is what is happening even in this gap. So if there's an action potential here, because there are no action potential here, the potential keeps decreasing. But if it is sufficient to cross the threshold here, then there's another action potential triggered. But now if you have two myelin sheets that are just removed, then suddenly the by the time the potential comes here, no, it's not enough to cross the threshold, and there's no action potential, and suddenly the propagation stops. And this is what happens during demyelination. So during demyelination, you basically have a small bit of the axon that gets depleted, the myelin sheath gets removed or attacked. For example, in multiple sclerosis, the myelin sheath gets attacked by our own immune system. So in such a case, this action potential triggered here, for instance, or triggered here, for instance, will the potential will decay because this is now no, there's no action potential here. So this is going to be a passive flow of, you know, current, or and this will decay. And if there are no ion channels here, or if the, you know, gap is long enough, then there is no flow of action potential, and suddenly you will not have, so your basic movements start suffering, because suddenly when you want to reach out and grab something, you can't, because the entire pathway is now hampered. Your brain might be functioning, your muscles might be functioning, but the gap, but the connection, the connecting cable between the two is faulty. uh, another interesting thing about this is, you know, there are a lot of naturally occurring toxins. So why are these important? Because these toxins again attack the underlying ion channels, which in turn will go and affect your action potentials. So in puffer fish, the ovaries of puffer fish, you have this chemical. There's this chemical called tetrodotoxin, TTX, which is a potassium channel blocker. So if you block the potassium channel, again, this, as we've seen in the cartoon, the action potential is not elicited anymore. In mamba snakes, you have this chemical called dendrototoxin, and this is also a potassium channel blocker, and these go and block in the nodes of Ranvier. So now imagine it that if there is a dendrototoxin acting here, and even if this thing kind of decays here, there's nothing for it to elicit an action potential. And this is not a toxin, atropine. I think we've all seen, come across this flower. This is this is the datura flower, and it's fairly common. And the chemical from here, the atropine, is actually used clinically to slow down your heart rate, because this one is an anti-muscarinic anticholinergic blocker, which basically means that at this junction, when this is stimulated and this one gets activated, this gap is blocked, this one will not get activated. So basically your initial depolarization itself is not happening, so let alone action potential. um, so here, this is what I've tried to show. So this is actually a simulation. So this is a Hodgkin Huxley model. This model is been around from 1950s, and this one has a sodium and a potassium channel, and this is a typical squid giant axon action potential. uh, and the minute you block any of the channels, the sodium or the potassium channel, you'll get no action potential. And these things can, and that's why they are quite fatal. But if you look at it in a different way, these things can, you know, blockers, channel blockers, and these things can be harnessed, and that's what they have also, in order to also treat ailments, or, you know, pathologies. For example, this procardia or adult cc, these comprise of something known as nifedipine, and nifedipine is a very potent calcium channel blocker. So this medicine is actually prescribed to treat patients with high blood pressure. How? Because if you remember the calcium, the once, if you remember, so this is again a model, the an atrial action potential. So if you remember this cardiac action potential, this bit of it is controlled by the calcium channel, mostly by the calcium channel. This peak is by the sodium channel. So if you block the calcium channel, basically this one, you're reducing the width, you're shortening the width of the action potential. And when you shorten the width and also reduce this, suddenly, as long as the potential is raised, the muscles contract, because when there's an inflow of calcium, we all know that calcium helps in muscle contraction. So once you reduce this, once you decrease the excitability and you reduce this gap, suddenly there's less muscles coming in and you end up relaxing the muscles. Another thing is that if any of you have ever struggled or had, you know, skin infection or fungal infection, this is one of the most commonly used medicines, creams. Or currently there are other creams, but this component is always there, clotrimazole. This is also a potassium channel blocker. So what it does is it targets the potassium channels of the fungus, not the skin, and it blocks it, thereby killing the fungus. Another one of another application, clinical application of a sodium channel blocker is lidocaine or exlocaine, which is actually used as a local anesthesia. So these things are injected into the body in localized places to induce a local anesthesia. And it's also used to treat ventricular tachycardia. So again, if we go back to this, so if we have, ventricular tachycardia is, a higher heartbeat than necessary. But if you block these things, and suddenly the gap increases between two consecutive action potentials, and thereby the rate at which the heart is beating also reduces. So during my doctoral research, my basic work was actually on bladder smooth muscles, and again looking at how certain ion channels would affect the electrical excitability of bladder smooth muscles. So there's not a lot known about bladder, and that's one of the reasons why we need to look at it, because if there is a bladder dysfunction, typically there are like blanket drugs that are given, which may or may not act. So there are no targeted therapies or things like that that cardiac muscles have. For instance, a heart, there are very chosen drugs that are given, and they have localized action, and they have been quite effective. But for the bladder, there's no control like that. Most of them are basically this anti-muscarinic, anticholinergic. So during my research, I had worked on one aspect of my research was working on a type of a potassium channel. And there I had simulated and seen that if this channel is activated in the case of spontaneous activity of the bladder, which is an overactive bladder, a bladder keeps firing every now and then, uh, and you are unable to control, and you just have the urge to micturate, then this activation of this channel, for instance, will help stabilize that. So this is just a glimpse of what I had done during my doctoral research. I wasn't sure if I should get into the details. I just thought I'll keep it simple. So in conclusion, in the field, in neuroscience or in any field, there are actually multiple levels, and each level actually provides a different set of information. And when you're looking to understand why an organ is behaving in a certain way, or why something has gone wrong, it's important to look at multiple levels, because you might be looking at the ECG, but ultimately the medicines that you go that is given to you goes and targets those ion channels right at the bottom. So when you are look so these things need to span multiple levels. You need to look at it at a system level, at a network level, and then at a molecular or submolecular level. But again, all of these things depend on what is the question you're trying to answer. So there are a lot of things that one could approximate while you're doing your research, or while you're doing your um, modeling or study. But ultimately, what is the question? So if you're looking at how a certain organ is behaving or responding to a treatment, injected or ingested treatment, then you need to look at all the levels, because all of these things would, when we inject something or ingest something in the body, all of them span the whole level. So thank you. And if you have any questions, and if there's not enough time, then please feel free to mail me. And if you have any questions, then please, I will be happy to hear.
So we have a few questions in the chat box. The first one is in the toxins slide. By blocking those channels, are the toxins not produced or just not released?
Oh, this one? Is this the one?
Yes, yes.
Hello?
Yeah,
Hello?
this slide.
This slide? Okay, no. So these toxins are, so for instance, if you come in, no, sorry, if anybody comes in touch with a puffer fish, their defense mechanism is to release the toxin from their ovaries. And when this toxin comes and hits any living creature, this is what the toxin does. So some of these, so if you look at, for instance, if the puffer fish goes and attacks a squid giant axon, a squid, sorry, a giant squid or a squid, and releases this tetrodotoxin as a defense mechanism or as a hunting mechanism, this toxin will go and block the channel, potassium channel in the squid, And as I showed in this particular slide, there's no action potential, and therefore no action potential would typically mean all there's an entire shutdown of the system. So basically, every organ has just stopped responding. So the creature is actually dead. So these toxins are not. This was the effect that if the toxin is given onto the cell. Is that clear?
Yes, yes,
Hello.
yeah.
Ah,
Thank you. Yeah. So I had also another question. So do temperature of the
mhm.
medium affect action potentials? Like for example, if we compare a normal scenario with in case someone is having fever, or other comparison is someone with hypothermia. So various organs act differently during those particular times in terms of action potential.
Yes. So action potential is not so much an organ phenomena as it is a cellular. So temperature, is a tremendous modulator of action of proteins. So the behavior of the ion channels will change with temperature. That's why it's very important that the core of our body stays the same. There's like a lot of things that within our body is, they're trying they I mean within I mean we don't know consciously, but within the body, there are a lot of mechanisms in place to ensure that there's homeostasis. So, yes, with different temperature, the action potentials will change. In fact, if you're interested, you can look up the temperature gated ion channels. So these are separate ion channels that actually modulate electrical responses based on the temperature. So at different temperatures, these ion channels get activated and therefore alter the response of the cell.
Okay.
TRPG.
Thank you.
Yeah.
So we have another question. Can you
Yeah.
elaborate a bit on the mechanism of the urge to control micturition?
Um, Mechanism on the urge to control micturition. So the thing is that the two different scenario here, that in a normal human being, your urge to control micturition becomes, there are a lot of feedback that you need, which is not necessarily only biological. So as your bladder is filled, you have um, afferent fibers that convey that information to the CNS, telling the CNS that okay, it's filled, it's filling, it's filling, it's filling, until a point you reach a maximum systematic capacity beyond which you can't control. Normal bladder, not a normal bladder, you just can't control beyond that. But till that point, when to micturate becomes a matter of social convenience as well. So suppose you are in a room where there is suppose you are listening to a class, you're in a classroom, and there's a test going on and you can't go, and you know that you want to go. So at that point, till you don't reach the maximum systemic capacity, you will go on controlling. So at those aspects, those parts, those centers of the brain will actually come into play, saying that okay, not now, not now, not now, and then maybe now. But in the case of a pathological bladder, what happens is that either the nerve, so there are multiple levels at which this can go wrong. So sometimes the nerve can be hyperactive, sometimes the muscles can be hyperactive. So what happens is that even when it is not full, there are signals that are sent. The bladder is probably, the muscle is probably continuously contracting, or the nerve is kind of, slightest contraction, and the nerve gets stimulated and says that okay, that it's time to void. So even when there's nothing to void, your bladder keeps sending signals that you need to void. And that is a pathological condition. Does that does that answer the is that is was that okay, or you want me to elaborate? If you do want me to elaborate, then let me know which part of it was not clear.
What exactly do we mean by pathological bladder here?
Pathological means um, something that is not normal. So something that's not functioning normal, that's a pathological bladder.
All right, all
So
right.
when you, so there are patients who have, bladder dysfunction. They are, overactive bladder or underactive bladder. The bladder just never tells the brain that, it's full and it needs to void. There
Right,
are things like that. So a pathological heart, for instance, a heart with, bradycardia or tachycardia, or atrial fibrillation or ventricular fibrillation. So similarly, malfunctioning bladder is what I meant by pathological bladder. That the bladder basically has two functions. One is it has to store, and the other one it has to release. But the thing is, it becomes so important because this is a matter of also, it has like a social connotation to it, that any one of these things, if the balance is not maintained, or if one of the functions are not done, the organ is not, it creates a severe psychological and physiological impact on the body.
right, right. All right, I got my answer. Thank you.
Great.
Does anybody have more questions?
I think we can move to the next part then, Swanzil.
Thank you.
Yes. Thank you so much for such an insightful talk.
Thank you so
And
much for the opportunity. And if anybody has any questions later on, my email ID is here. Please feel free to reach out.